Vector Builder is an online bioinformatics software and database for building and documenting DNA constructs. It gives you a drag and drop plasmid construct editor, a library of common genetic parts like promoters, tags, markers, origin reporters, and also tools to annotate sequences and ready to use maps and files. You can design cloning strategies, check key features in the sequence, and keep the projects organized with clear records of what each vector contains. It is a short and one-stop platform to design, visualize, and manage vectors more efficiently for molecular biology work.
In the biotechnology field, Vector Builder addresses a lot of practical and industrial challenges beyond basic molecular cloning. Firstly, it supports bioproduct development. Biotechnology often focuses on producing proteins, enzymes, vaccines, or metabolites. Vector Builder helps design expression vector vegetable promoters, signal peptides, and tags. Therefore, it improves expression efficiency in animal, plant, or mammalian systems. Furthermore, it accelerates strength and cell line engineering. Vector Builder allows researchers to plan gene injections, knock-in or knock-out strategy clearly. Thus, it reduces development time for production strength. In addition, it reduces the cost and material waste. Field cloning in biotechnology can be expensive due to specialized reagents and long culture times. By simulating contrast in advance, Vector Builder minimizes unnecessary experimental repeats. In short, in the biotechnology field, Vector Builder enhances productivity, reliability, and scalability. It supports efficient bioproduct development, synthetic biology, and industrial strain engineering while reducing cost and experimental risk.
In molecular biology, vector builder can be used to solve multi-gene coordination. There is sometimes a need to express three or four different proteins, but this plasmid doesn't always enter the same cell. Therefore, vector builder can be used in this situation to solve this problem. This is the demonstration of how the software can be applied to solve problems. So firstly, the vector is selected, followed by selecting the number of RFs. We can select 2 or 3 here. This can be understood as number of genes. A strong non-silencing promoter is then selected, followed by the other selections. This can be selected or copy the sequence that we have obtained, then add to A linkers. Finally, press the marker with antibiotic resistance is selected, such as EGFP. ORFs is then selected using the DNA database or uploaded from DNA sequence obtained from experiments. Finally, press Finish Design to see the result vector.
Next, I'm comparing the usefulness of Vector Builder and Invitrogen for vector construction. Vector Builder is a custom vector design and synthesis service. Its main strength is high customization and reliability. Researchers can design complex plasmid or viral vector online, and the final product is delivered sequence verified and ready to use. This saves time and reduces experimental errors, but it is more expensive and offers less hands-on learning. In contrast, Invitrogen provides ready-made vector, cloning kits, and reagents. It is more cost-effective for standard experiments and offers strong learning opportunities because researchers perform the cloning themselves. However, it is less suitable for highly customized or complex vector and may take longer due to manual lab work. In conclusion, Vector Builder is more useful for complex time-sensitive projects, while Invitrogen is better for routine experiment and skill development.
Vector Builder is an online platform that allows users to design customized vectors and directly proceed to vector construction and production. It is very useful because it combines design and laboratory services in one workflow. In comparison, Vector B mainly focuses on in silico vector design. It allows users to edit DNA sequences, annotate vectors, and plan cloning strategies. It is free and useful during the early planning stage of experiment. However, Vector B does not provide vector constructions or production services. Users need to use another service to build the vector, which can take more time. Overall, while Vector B is helpful for design and analysis, Vector Builder is more useful for complete research workflow because it saves time, reduces extra steps, and supports users from design to final vector productions.
In conclusion, Vector Builder is an effective for demonstrating multi-gene coordination because it supports designing and organizing complex constructs in one workflow, which speeds up bioproduct development and also improves efficiency. Compared to in vitrogen, Vector Builder is typically more focused and on an integrate design and build platform rather than mainly individual cloning products. Moreover, if compared to Vector B, Vector Builder generally offers broader support for more complex multi-gene products, and overall it is a strong choice when the priority is efficient multi-gene construct development.